Welcome to part 1 of 4, looking at chapter 11. This chapter we're going to be starting looking at DNA and genetics in the next 2 units. So chapter 11 we're going to talk about the nucleic acid structure, we're going to talk about DNA replication, and then we're going to end up with chromosome structure. So this unit, chapter 11 and 12, really focuses on nucleic acids, which if you remember from chapter 3 is one of our 4 types of macromolecules. So we have talked a lot about the carbohydrates, we've talked about lipids, we've talked about proteins quite a bit. So here we're really getting into that 4th nucleic acid group of molecules. So one type of nucleic acid is DNA. And DNA, this is the genetic material that provides the blueprint to produce an individual's traits. So the DNA is basically all the instructions in the cell that give us all of our traits and characteristics and help cells decide what type of proteins they should make, what the cell should look like, how many cells an individual should have. So it's really, really important to have this genetic material, these instructions, in order to grow and reproduce living things. So DNA was discovered in the 1950s, so it's actually fairly recently in terms of how long people have been looking at living things that we have actually discovered that DNA codes for all these traits and characteristics. So I'm going to quickly go over identification of genetic material in this part of the chapter. The other 3 parts, part 2 will look at molecular structure of DNA, part 3 will look at DNA replication, and then part 4 will be pretty short, looking at chromosome structure. So how the DNA folds together, wraps up to fit into cells. So identification of our genetic material. Before the 1950s, people recognized that there had to be something in cells or something in living things that coded for our traits and characteristics. So they came up with criteria of what this molecule should have, or what properties it should have. So they decided that this genetic material, it had to be able to hold information because it had all the information to give us our traits and characteristics. This genetic material, it had to replicate itself in some way because it has to be passed on from one generation to the next, which is that transmission part, so passing it on to offspring. And then in addition, this genetic material had to account for all of the variation that we see both within species. So if you think about looking at different humans, we have a lot of variation in humans. And then also this has to account for variation between species, like how humans were built much differently. We have different traits compared to like dogs, or compared to birds or reptiles or bacteria. So we have all this variation of different organisms that we have to account for. In the quest to identify this genetic material that has those 4 criteria, researchers started looking at cells using the microscopes that were developed at that time. So in the 1800s, researchers became convinced that the chromosomes carry the genetic information. So what happened is we have this really simple experiment that we're going to look through. So if you remember prokaryotic cells, which are those simple cells that don't have a nucleus, prokaryotic cells, they're basically a cytoplasm with some ribosomes in there and then one circular chromosome. So it makes sense that scientists would use a prokaryotic organism, specifically a bacteria organism, in order to look and see if chromosomes are actually carrying genetic information that coded for traits. So in the late 1920s, we have Frederick Griffith, and he was working with a species of bacteria called Streptococcus pneumoniae. So this species of bacteria, it has a prokaryotic cell, so it's basically a cell with that one circular chromosome inside of it. So this Streptococcus pneumoniae bacteria, it comes in 2 types. So there's a smooth, or the S type, and this is a type that will cause pneumonia, which will be a fatal infection. So people, or he worked with mice, became very, very sick, and generally they would die from this infection. But besides the smooth type, there's another type called the rough type, or the R type. And if an organism gets infected with R type Streptococcus, they're able to survive, so it's not fatal. So he was looking at these two different phenotypes of the same species of bacteria. So what he did was that he set up two controls. So he had this bacteria growing in the lab. He wanted to test if his strains actually did what they're supposed to. So he took mice, and treatment number 1 was a control. He injected mice with that S type, that fatal type, just to see that his strain was actually fatal to the mice he was working with. So he took this S-type bacteria, injected it into a mouse, found out that the mouse died from it because it got sick and it's fatal. His other strain, the R strain, he also injected that into a mouse to check to see if it would do what it's supposed to do, and he found out his R-type bacteria, he injected it in, and the mouse lived. Okay, so this again is just those controls to show that the S-type is fatal, the R-type is not fatal. So then what he did next is he took that S-type bacteria, the type that is normally fatal, and he heat killed the bacteria. So he tried to destroy the bacteria, destroy the DNA. He then took that heat-killed S-type, injected it into a mouse. That mouse survived. Okay. So this shows that you can kill that bacteria and you can stop that infection from being fatal. And then his fourth treatment, this is a really important one. So far we've just kind of been doing controls to make sure that our bacteria are working how they're supposed to work. So in his fourth treatment, he took that living R-type that does not cause an infection, he mixed it together with the heat-killed S-type bacteria, which in treatment number 3, if it's heat-killed, we know it does not cause an infection. But when he mixed these two types of bacteria together, the mouse died. It got pneumonia and died. So what he concluded from this is that that heat-killed S bacteria, there's something in there that got into the R-type and turned it into the fatal strain. So you can see he collected bacteria from the mouse that passed away, and he found that the bacteria in the mouse was an S-type bacteria. So he concluded that the living R cells, they've been transformed into the fatal S-type cells. So there's something that was transferred from one strain of bacteria into the other strain of bacteria, and that it made that bacteria fatal. So if we kind of review these 4 criteria, we can match them up with Griffith's experiment. So remember, we have to have some molecule that contains information, it has to be replicated, it has to be transmitted, and it has to account for variation. So in relation to these 4 criteria, in this experiment, we have our genetic material from our heat-killed type S bacteria. It was transferred into the living type R bacteria. So that matches up with criteria number 1. So this genetic material, it holds information in order to make that bacteria strain fatal to the mice. The trait that was transferred gave that bacteria a capsule, which causes it to be fatal. So we match up with number 1 and 2. So the genetic material holds information, and then that genetic material has to be replicated. That genetic material is also passed on to the bacteria offspring, which matches up with criteria number 3. It's transmitted. So we have these chromosomes in our prokaryotic cells. They match up with the first 3 criteria. So chromosomes, they contain information, 4 different traits. That information is replicated, and then it's also transmitted to the offspring. So we have these chromosomes that fit really well in with these 4 criteria that we're looking at. Okay, so we have this chromosome that carries this genetic information. The problem with chromosomes is more scientists work with chromosomes, they realize that chromosomes are made up of two different types of molecules. So chromosomes, they have protein part to them, and they also have a DNA part to them. So the interesting thing with this is that proteins, there's 20 different amino acids that make up proteins, so there's a lot of variation of proteins. DNA only has 4 nucleotides, so fewer different types make up the DNA. So researchers are really convinced that protein was actually the genetic material, because it would account for all of that variation that we see within species and between species. So in order to decide if it's the proteins or the DNA that carried that genetic information, we have a really big experiment by Hershey and Chase in 1952. So luckily, we have things called viruses that are made up of proteins and DNA, which are the two things that we're trying to decide between as our genetic material. So there's a specific type of virus called a bacteriophage. So this is a virus that will infect bacteria cells. So we took, or I guess Hershey and Chase took a bacteriophage. They took a virus shown on the left-hand side. It's a T2 bacteriophage. This bacteriophage infects E. coli cells, so bacteria cells over here. So what happens is that they wanted to determine if the protein part of the virus was being put into the E. coli or if the DNA part of the virus is being put into the E. coli. Because viruses, they basically will hijack the cell, take it over, and use that cell to make more viruses. So they really wanted to know which part of the virus was being put in the E. coli, so which part of the virus was taking or hijacking that E. coli cell. So they did this experiment to figure out what was being injected into the E. coli bacteria, the DNA or the protein part of the virus. So they took a virus, and one tube they labeled the protein part of the virus with a sulfur isotope, so S35. In another tube they labeled the DNA with the P32 isotope. And then those two separate tubes, they just used a blender, so very simple, to separate the bacteriophage from the bacteria once they were mixed together. So here's kind of what happened up here. So in experiment number 1, this is where the protein was labeled with our S35 isotope. So we had E. coli cells, they were infected with our S-labeled phage, and then we put them in a blender to shear the phage off of our bacteria cell. They then took that tube out of the blender, put it into a centrifuge. The centrifuge, it causes the bacteria cells to go to the bottom of the tube and all of the liquid to stay up above the bacteria cells. So they found that the S-labeled protein part was in the supernatant, which is the liquid. The E. coli cells at the bottom of the tube, they were unlabeled. So this is showing that the protein part of the virus stayed in the liquid and it wasn't found in our bacteria cells. Okay, so the protein's not going into the bacteria cells. So just to kind of show that it's the DNA going into the bacteria cells, they did experiment number 2 over here. So kind of the same thing, they put E. coli cells in with the virus, except they labeled the DNA part with a different type of isotope. They put them in a blender, so the virus separated from the bacteria cell. They put it in the centrifuge, so the bacteria cells went to the bottom of the tube. The viruses stayed in the liquid part of the tube. And then they looked for that isotope. And they found that the labeled viral DNA was inside the bacteria cells, right here. So this is showing that the viral DNA was being injected into the bacteria cells, and the DNA was the part that was hijacking the bacteria cells to make more viruses. So from this we conclude that DNA is the genetic material. It's not the protein part of the chromosome, it's the DNA part of the chromosome, which we're all familiar with nowadays, but at the time this was pretty big and exciting because everyone was really convinced it was the protein part and not the DNA part. So because DNA is the genetic material, we're going to spend the rest of this chapter looking at DNA, how it's structured, how it's replicated, and then how it's actually packed into those chromosomes that we talked about at the beginning of this section in this chapter.